So when it comes to type 1 diabetes, as you probably know, it's an autoimmune process of destruction of pancreatic beta cells, and incidence and prevalence of type 1 diabetes vary significantly. So this prevalence and incidence actually are rising all the time. And what is also very interesting with type 1 diabetes, that actually the prevalence is very huge so the rate of the incidence is also huge in developed countries. So this is something that is not characteristic of many other diseases. We don't know actually why this autoimmune process starts in type 1 diabetes. Viral infection is something that is maybe supposed to trigger it, but it's not for sure. But we know that both genetic and environmental factors together play an important role in predisposition of type 1 diabetes. Previously, diabetes type 1 was considered to be related to child population, but here we have the actually data from 2022, which says that 62 newly diagnosed cases were actually in people aged above 20 years or older. So it is also important to mention that many people with type 1 diabetes develop complications such as chronic kidney disease, diabetic peripheral neuropathy, etc. So now we saw that actually diabetes is the disease with many open clinical questions. But then after that, you must actually ask yourself a question: can your molecule, your approach possibly offer a solution? So I just put all this on one slide, so the glycobiology on one slide, because I assume that all listeners are from the field of glycobiology, but everything we know about glycans today actually supports the possibility that glycans could be very valuable biomarkers. We know that majority of molecules in our body are glycosylated, that glycans modulate protein functions, that the process of glycosylation is very complex, strictly regulated, and actually that glycosylation is regulated by the network of genes, so the huge number of genes, and not only those directly involved in the process of glycosylation. So actually, each point is potential target to us, because the disruption in each step can be actually the trigger for some kind of pathophysiology. We also know that although glycom show great inter-individual variation, that N-glycome is stable within an individual during the period of homeostasis. We also know that N-glycome is changed in many diseases, and that glycans actually change with great sensitivity, and that this change occur very early in different pathophysiological conditions. This is why we know that our potential marker, thus glycans, have good potential in prediction, stratification, and differentiation of many diseases. What is also important that from our studies and studies from others, we know that N-glycosylation is actually included in diabetes development. For instance, some genome-wide association studies identified glycosyltransferase genes as candidate causal genes for both type 1 and type 2 diabetes. Those were, for instance, FUT2 gene as causal candidate gene for type 1 diabetes, as well as TES6-GAL1 as a novel locus implicated in the development of type 2 diabetes. What we also know that glycans have a major role, for instance, in the proper orientation of glucose transporter, as well as the retention time of T cell receptors on cell surface. So we know that actually the changed glycosylation influence these processes and can be involved in the development in both type 2 diabetes as well as the autoimmunity in type 1 diabetes. On the other hand, for instance, when it comes to monogenic diabetes, in our work, we showed that glycans are really promising biomarkers for monogenic diabetes, since in our studies on HNF1A MODY, it's a maturity onset diabetes of young, so diabetes usually occur in young adults, but is often misdiagnosed as type 2 or type 1, and is caused by mutations in an HNF1A gene. So we showed that glycans are changed in patients with HNF1A modi. Actually, it was the glycosylation which was really lowered. But the current diagnosis of this type of MODI is actually established in a way that you need to sequence the whole gene. And then if you find, for instance, a novel mutation, you must conduct functional studies because not all mutations are pathogenic. And glycans actually substituted this whole process because what we show that glycosylation was lowered only in people with the disease. So only in people carrying the pathogenic mutations, but not in those with the benign one. Also, when it comes to diabetes type 2, we know that diabetes type 2 is a huge epidemiological problem. And what we also showed in our work is that glycans change even 10 years before the diabetes development, and that this change actually progress as towards the clinical symptoms of the clinical manifestation of the disease. So now, when we know that actually glycans are good potential biomarkers, that we know that they're involved in the process of diabetes development, and we decided to explore their biomarker potential in type 1 diabetes. When we started our research, there was no study actually that plasma protein glycosylation in patients with type 1 diabetes. So we were lucky that due to our collaboration with Steno Diabetes Center from Copenhagen and the University of Western Australia, we got into possession of almost 2,000 samples from children with a new onset type 1 diabetes. The subset of 188 children also had 244 healthy siblings sampled. So what we did, we analyzed glycans in both 2,000 children with type 1 diabetes and this subset of healthy siblings. We used our usual approach includes enzymatic glycan release, labeling, and HILIC-UPLC analysis. And we separated glycans into, when it comes to total plasma proteome, into 39, and when it comes to IgG glycans in 24 glycan groups. So when we compare the glycosylation between children with type 1 diabetes and healthy children, we actually identified many glycans that were different in two populations. Here in the red are actually the summed features of these glycans, and what you can see here that the features that differ the most were bisecting, monogalactosylated structures, and high manostructure. And on IgG, we also saw the difference, let's say the increase in desilylated structures. So for us, it all made sense because decrease in monogalactosylation was previously also connected with other autoimmune disease. And when it comes to increase in high manos and bisectin, which we occurred, we said, okay, maybe it has to do something with increased binding to MBL-lectin, and we know that actually complement system plays role in type 1 development. Increased cellulation of IgG could also be connected to inflammation. And here it was also maybe interesting to us that this desilylated glycans was actually previously reported or assumed to mainly come from FAB region of IgG. So now we are actually trying to see if that was the fact. And recently we took the subset of the same samples from the same cohort. We used EDAS enzyme to separate FAB and FC region. And actually we identified which glycans are present on which part of the protein. And when we compared glycans on FC region and FAB region, in children with type 1 diabetes and healthy siblings, we saw that the majority of differences we observed in our population on total IgG are actually originated from FAB glycans. This is something we are still working on and still need to actually explore our results a bit further. So, but before that, we actually used our results from total plasma protein N-glycosylation and IgG glycosylation, and actually constructed a discriminative model in predicting diabetes status of patients with type 1 diabetes and healthy controls. And we saw that glycans possess really good differential potential. So you can see for plasma glycom, we got area under curve of above 0.9, and for IgG, it was almost 0.9, so 0.86. So then the second question arises. Okay, now we saw actually which glycans are changed at the beginning of type 1 diabetes, but what is with adults with type 1 diabetes? Do their glycom change further? And we saw that when we compared N-glycom profiles from adults with type 1 diabetes and healthy controls, we saw that their glycom actually exposes further changes compared to children at the disease onset. So we identified some glycans that were common to adults and children with type 1 diabetes. And for this, we can maybe assume that they are somehow connected with genetic factors that contribute to the predisposition of development of type 1 diabetes. We also saw some changes that are specific to adult subjects, so maybe they reflect the altered glucose metabolism or actually some inflammatory processes going on in these patients, or the development of some complications. And also there were some changes unique to children, and these are maybe also very interesting one. We still don't know why they're there, but maybe those are actually potential targets for intervention and monitoring of early disease progression. So we didn't only compare the glycoms between children with type 1 diabetes and healthy siblings, we also used this large cohort to actually see which genes regulate the glycosylation of total plasma proteins and IgG. And since these actually kids had immunochip done. So we conducted genetic association study on N-glycosylation in type 1 diabetes, and we got a few hits, few associations, but here are some that were actually novel. So different from previous GWAS studies done on the genetic regulation of glycosylation. Those were hits, as you can see, mainly connected to this process of glycosylation itself. But the one that was very interesting to us, and that was the connection, the association between the C3 gene, so the gene for component complement-free, and oligomannose glycans. Here you can see these results. And it is also important to mention that actually one of these SNPs is missense mutation. So we have the change, as maybe you can see it better here, of the amino acid being changed from proline to leucine. So why complement component 3 was very interesting hit to us? Because the C3 is actually the central and most abundant protein of the complement, and it was already genetically associated previously with type 1 diabetes. It was also associated with diabetic nephropathy, and from the side of glycobiology, we found a paper that was done only on a few samples, but it reported that a C3 protein actually bears only high mannose glycan structures, which is not usually the case for plasma proteins. And I think that majority of also components of complement system carry complex glycans, such as other proteins in our blood. So it is also important. It was also interesting to us that oligomannose glycans glycan with 9 mannose units that we found to be associated with SNPs in the C3 gene is actually attached on domain involved in C3 binding with pathogens. So this is the TET domain that is further exposed after the activation of C3 protein. So we wanted to explore the C3 glycosylation in children with type 1 diabetes, but for that, we first needed to develop high-throughput method for C3 N-glycosylation. So here you can see the method that the approach, the protocol that we developed, which actually includes the enrichment of high mannose glycoproteins using con-A affinity capture matrix, and then the subsequent LC-MS analysis of glycopeptides, which offer site-specific C3 N-glycosylation analysis. So after we used this method on plasma samples, we saw that C3 protein actually has two glycosylation sites, which are exclusively occupied with oligomannose glycans, both sites. And we tested our method for reproducibility, and we saw that it's pretty reproducible for all glycoforms. So then we proceeded with using this method to analyze C3 N-glycome in children with type 1 diabetes compared to healthy siblings. And we saw that there are differences in its glycosylation. And from this box plot, you can see that actually on both glycosylation sites, so this is the one and this is the first and this is the second, we can see the increase in less processed oligomannose glycans, so glycans with more mannose units. And what was also very interesting, that on the second glycosylation side, we also saw the oligomannose glycan with 9 mannose units and glucose attached to it, which is really rare. So we constructed C3 N-glycan discriminative model and also saw that actually C3 N-glycome can actually discriminate between children with type 1 diabetes at the onset of the disease from their healthy siblings. What we also wanted to check, as I already told you, complications in diabetes is something that is very important clinical questions. So we also analyzed C3 N-glycome in type 1 diabetes related complications, and we saw some changes with associations with albuminuria and hypertension. But I must also stress here that actually we only had individuals with mild complications, so maybe some additional studies has to be done. But here we can see that actually the C3 N-glycome can also maybe carry potential as the prognostic biomarker in this manner. Actually, all the results I presented just now open new questions for us, and we are just in the process of answering them. So actually, as you saw, the glycosylation of total plasma proteins, IgG and C3 occurred or actually were present at the beginning of the disease in children. But still the question remains how early these changes occur in the course of the disease. So it would be great if we can prove that these changes actually precede clinical symptoms. Can they be then marker for at-risk children? So the second question was, okay, now we show that C3 glycosylation is changed in children and adults with type 1 diabetes, but what is the actually normal referent glycosylation of C3 in general population? Is it age and sex dependent like other glycoproteins that we studied? And which genes actually regulate its glycosylation? And maybe the most important when it comes to the point of type 1 diabetes, what is the role of N-glycans for C3 function, and does it have any implication to type 1 diabetes? So actually, as I said, we are now trying to answer all these questions. So I will show just the preliminary data for you to see how we are doing it. So these questions will be answered through the project that is funded by the Croatian Science Foundation, and is called Glycosylation of Complement Component C3 in Development of Type 1 Diabetes and Autoimmunity. On this project, we are collaborated with Clinical Hospital here from Zagreb, Merkur, as well as the University of Adelaide and the University of Western Australia. So the first question, the question of how early in the course of the disease these glycosylation changes occur, we will try to answer using this great Australian India study. It's the environmental determinants of islet autoimmunity study. So actually, the study sampled newborns who had either mother, father, or sibling with type 1 diabetes. The study actually followed mothers from pregnancy and then sampled children at birth and followed them at every 3 months, and they're still following them. So actually the study is still ongoing. Till today, the study is actually designed as the study that the endpoint is still not the type 1 diabetes, but the presence of islet autoimmunity. So the cases are actually children who developed islet autoimmunity, so who has proven presence of autoantibodies in their samples. The study is organized in nested case control manner. So we received actually 51 samples from children with autoimmunity and 161 controls. And the study is designed that each case has 3 controls matched in a separate nest. So since this approach actually demands some more complex statistical methods and approaches, our statistician designed, together with the statisticians from India, designed some longitudinal models to actually follow the behavior of glycans in children from birth to the month of 60 months in both cases and controls. So these are actually results that we are exploring further now, as well as we are actually trying to see in each time point whether glycans differ between children with diabetes and with autoimmunity and healthy children. And these are the graphs from models constructed for that. You will probably understand them more than I do. And what is the point here? That these dots represent actually the statistically significant differences, and the lines represent 4 different models. The lines are the blur because the more transparent the line is, there are less samples in that time points. So actually we are here following children to the onset of autoimmunity. And here you can see that when it comes to some of C3 glycans, there are statistically significant differences between children with autoimmunity and healthy controls for all designed models at the time of the autoimmunity development. And for instance, here, when it comes to plasma proteins and IgG, here is this those high mannose glycans that I said are really interesting to us. And from here, you can see that these glycans are changed at the onset of autoimmunity, but also change 3 months prior to the development. So this is something that we are still working on. The second question that we are still working on is the question of N-glycosylation of C3 in general population. So we actually analyzed C3 N-glycomes in 816 adults from the Croatian island of Korčula. And here you can see that, for instance, contrary to IgG, whose glycans are really age-dependent, it is not the case when it comes to C3 glycosylation. So C3 glycosylation is pretty stable through the lifetime in adults, but there are some sex differences, so differences between men and women, but they are not so expressed only for some, let's say, glycoforms. But this is also maybe good from the point of potential biomarker. Since then, this biomarker is, let's say, more robust since it is not that influenced by the age, so you must not divide people in some age groups when considering it as a biomarker in for certain disease. What was also really interesting to us when it comes to C3 glycosylation is that when we associated or correlated, let's say, different C3 glycoforms with some biochemical features, let's say other phenotypic characteristics, and we saw, and age and sex adjusted, we saw we saw connections shown here. But then when we actually adjusted it further for BMI, many of the correlations were lost, but it was very interesting to us that features such as glucose, lipidogram, HbA1c remained. And it was not usually the case for other glycoproteins we were studying, because usually those were connected with the BMI. So here we can see that N-Glycome from C3 actually add some information and is probably important in this metabolic status of an individual, which supports the fact that it may also be important in development of different diabetes subtypes. So what we also saw from our study, we saw actually that what are the levels of each different glycans on each glycosylation site. So here we can see that on the first glycosylation site, this oligomannose glycan with 6 mannose units is the dominant one, and the glycan with 9 mannose units is the dominant on the second glycosylation site. And the question remains why C3 glycoprotein only bears highly mannose glycans. So why doesn't it has complex glycans also attached? And Oliver Grant from the University of Georgia is helping us to actually answer this question. So he modeled some accessibility. He actually made some accessibility models which show that mannosidase that is supposed to further trimmer mannose units from those glycans can't really, or that these two glycosylation sites are pretty inaccessible, or let's say, yeah, so this the rate of the trimmer is very, very low. So we also use this general population data and C3 glycosylation to study the genetic regulation of C3 N-glycosylation, and this is something that we are still working on, but for now we discovered a few associations, and if you look through the list, those are mainly some genes connected either to the process of glycosylation or the autoimmunity or some immunological features. So this is all from type 1 diabetes, but what I also wanted to tell you, it's not connected to this project I just showed, but it's another project that was just funded by the EU development funds in our lab. And this project actually includes collaboration between industry and academia. So apart from our faculty, we have the collaborators from Zagreb Center for Gut Microbiome, Faculty of Food and Biotechnology, and in silico Ltd. But this is the project that will study and will combine glycome and microbiome as markers of health in women during reproductive age. Why I put this here? Because we are just starting the research in this field, so maybe it would also be interesting to some of you, and maybe we can also somehow collaborate on this topic. I would like to thank to all our partners, to our funding agencies that are supporting us, and I hope that from my talk I just chose the small part of our results, but I just wanted to actually show you how we think and how we explore the diseases and biomarker potential of glycans. So thank you very much.